I feel like I ended the other vodcast a little abruptly and did not give the code its due time because I was worried about how long the talk was going. So I'm going to recap a little bit of that and also remind you that we are in chapter 10. After I uploaded to YouTube, I realized I think I said chapter 11. So we are in chapter 10. And just want to talk more about the code because it is a pretty fascinating thing. And one of the um, huge ideas to remember is that it's actually conserved all the way down to the most simplest bacteria. Every organism uses the same genetic code. If you specify CAC and you are in the most primitive bacteria, or if you're in humans, you will still code for a histidine. And that's pretty enormous. There's one or two species that they've found so far that have a few alterations, and they just seem to be some experiment that didn't work out. But basically, the code is preserved. And what we're going to find out when we go into the molecular biology, more technology unit that's coming up, is that that'll allow us to put the mRNA from a human species or a mouse or a jellyfish, for example, and put that mRNA into a bacteria and have the bacteria make that protein for us, which has turned out to be a pretty enormous tool in biotechnology. We produce some drugs like that, like insulin. We produce some products that we use just in food processing, perhaps, and enzymes like that. And it really comes out of this code. So again, going back to the idea of how they discovered it, the code was ready there when Watson and Crick figured out the structured DNA and that's how it was copied. They identified, they meaning scientists, identified RNA and realized it was a complement to the DNA. And then finally they needed to decipher what that RNA could code for. And I told you about how the different combinations, if you took them 2 at a time, you would only have 16 possibilities. Remember, that's a square. And if you take the bases 3 at a time, which is called a triplet, we can have 64 possibilities. And that's what we have here. Again, these are the amino acids, the whole words. You'll see them sometimes abbreviated just as a first 3 letters. And there's a single letter code. Sometimes it gets used. You can see that some of them, like proline, for example, have multiple ways you can code for it. Leucine, look at that, 6 of them. What that allows is for redundancy. It allows for the first 2 bases to be conserved, and then any change in the third one allows you to still code for the same amino acid, which is really awesome. We want to absorb mutations and not have them change our protein sequences, and then sometimes you do want to have the mutation change the sequence. And that's the topic of the next little bit here, is how can all these mutations result in changes in our proteins. So there's 4 main kinds that you want to think about. Silent mutations, meaning there's no change in the protein. Missense mutations, meaning we swap an amino acid code. Nonsense mutations makes a stop codon, so we turn into one of those 3 stop codons that are in the code. Or a frame shift mutation, and that's kind of the coolest one. It's when you insert or delete a base, you can shift the way you read the triplets. And I'll look at them each specifically here. So a silent mutation, that's the one where it's in that 3rd position of maybe an amino acid that gets coded for by multiple codons or multiple triplets. So again, the original sequence was an A, something happened, and you swap it out and you get no change in the amino acid, no big deal. The missense mutation is when you had mutation turns from into an A instead of a T, that results now into a U here. They're not showing us the original strand, they're showing the mutated strand. And then that codes for a new amino acid, valine, instead of what the other one used to be. Used to be an aspartic acid. So just by changing one nucleotide, see this one's in the 2nd position, so it's going to be more dangerous, we get a whole new amino acid. This one could probably be an interesting change for the protein because aspartic acid has a charge on it, and valine is a non-polar amino acid, because now you might be disrupting the structure of a protein. You have to think about what the downstream effects are on the protein there. Continuing on. Nonsense mutation means that you introduce a stop codon. So again, they're saying here that you swapped a T in here instead of what used to be a C. So now the mRNA is going to read UAG, which is a stop codon, which means that you're not going to make a protein. Anytime that happens in the protein, you basically kill it. One thing to think about with mutations, if the mutation happens in the germ cell and the DNA gets changed, then you're going to pass that down to your offspring, right? But what if this is a mutation when the skin cells were copying themselves, making a new skin cell, and this mutation only exists in that new skin cell? What are the implications of that? Well, that means that that skin cell won't make this protein because you have a stop codon in there. Will that be dangerous? Maybe, maybe not. Again, it's that one cell. Maybe that one cell will die. But what if that mutation makes that one cell, and maybe this happened in a proto-oncogene, remember those? And now we're turning our proto-oncogene into an oncogene, and now maybe that skin cell is going to grow and grow and grow and grow. So there's all kinds of implications about when and where the mutations take place and what the implications are for the organism. Something to kind of wrap your head around. Frame shift mutations. I love these. I think these are the most interesting ones. So in this case, they're saying that you had a sequence that read CCG, and now you've inserted a T for some reason. There was an accident in replication, excuse me. And now we're going to read in triplets. We're going to read this one, UAG, is a complement of TAC. Then we're going to read UGG, which is a complement of ACC. Everything's good. But now, instead of reading the GAG, which should have been there, we're going to read it TGA, which translates in the triplet of ACU. Voila, we have a totally new protein the rest of the way down. Usually when frame shifts take place, it's just a fascinating thing that when you are out of frame, you're not in the correct frame, you start hitting stop codons. They're not showing you the whole thing here, but in nature that seems to be what happens. When you're not reading in the correct frame, you tend to hit stop codons. It's kind of freaky. What they're showing you here is that you're going to add in different amino acids, which is what happens a lot of the time. If you drop in a stop codon, again, garbage, gobbledygook. But this is a pretty wild mutation. So I'm going to stop, well, and maybe I'm just going to keep going, and we'll start talking about tRNA here. We'll call this one mutations in tRNA. So now we're going to get ready to decode our mRNA. So we know that the DNA gets turned into RNA in the nucleus. We know that the RNA is going to get processed. Whoops, don't want that. Okay, don't complete your backup. There you go. We know that the RNA is going to get processed. We are going to remove the introns and splice the exons together. So this would be exons put together. We're going to put the 5-methyl G cap on, and we're going to put the poly A tail on. And now this mRNA is going to be allowed to get out to the cytoplasm. So what's going to happen to that mRNA there? Well, it's going to end up meeting up with, I'll jump ahead to this picture, where it's going to meet up with the 2 subunits for the ribosome that you guys heard about ages ago, and I told you we'd learn more. But let's first talk about how, when it's being read, something is going to deliver the amino acids to these sites. So here's our first triplet, AUG. So remember that codes for a methionine. So we need to have something drag a methionine over here to match up to that mRNA. And here it is. So tRNA, or transfer RNA, is the type of RNA that, so it's called tRNA. This is a type of RNA that will deliver our amino acids. So remember ages ago, again, when we talked about macromolecules, we said there were all different kinds of RNAs. So there's tRNA, there's mRNA, we're very familiar with that. We're going to talk in a second about what the ribosome is made of. It's made up of rRNA. In the next chapter, you're going to learn about small interfering RNAs, or siRNAs, also a similar idea, but gives another name. It's called RNAi, or RNA interference. They're both the same idea. They're interfering with the RNA. And there's probably some more. So we're just going to focus right now on tRNA. tRNA gets this name, believe it or not only because it's a transfer, but when they first figured out some sequence information, it kind of looked like a T, handy. The tRNAs are all built like this. You have at least 20 of them because we have to deliver 20 amino acids. So the tRNAs have a 3' end and a 5' end. They're single strand of RNA, but then they get the superstructure because they can hydrogen bond. See how that G and the C and the G and the C and the A and the U, blah, blah, blah. And when they do that, what in the world is a D? Is that a typo? Huh, crazy stuff. Anyway, when they make this, they make it into a superstructure, and that's what RNA can do. The important end is, there's 2 important ends. Down here, is called the anticodon. So this is the one that's going to match up with the codon in the mRNA. So this is the cognate thing. It's going to identify which amino acid should be bringing over. And then again, the other business end of this molecule is the 3' end. It's where you always, it's CCA always, and it's where you attach the appropriate amino acid to this guy. When they did the crystal structure of these, remember we can crystallize it and bombard it with x-rays and find out what it really looks like. Unfortunately, it doesn't look as much like a T, it says L shape because it actually bends, one of these guys bends over on itself and it doesn't look quite like a T. In any event, still has the business end down here with the anticodon and the other business end up here where it's going to attach to an amino acid. How does it get that amino acid? Well, there's an enzyme that does it, of course. The enzyme is not shaped quite like this. This is a little bit too much of a cartoon rendition, but it gives you the idea. So you have an amino acid tRNA synthase for every single amino acid. We have 20 amino acids, so there's a site to bind that specific amino acid. In this case, they're showing you leucine. There's a site to bind your specific tRNA. You use up an ATP, voila, you attach your amino acid to your tRNA, and now this is a charged tRNA ready to go over and build a protein. So think about all the energy that takes place in building the protein. I'll bring this up a little bit later. Everything costs energy in the cell, right? In the terms of ATP. We're going to bind our amino acid with ATP. So for every time we deliver an amino acid over there, we've already used up an ATP. All this is floating around in the cytoplasm. As soon as this guy goes and drops off his amino acid at the ribosome, it goes and bumps into another enzyme, which finds a floating leucine around from maybe when the proteasome digested up some other protein and dumped off its amino acids in the cytoplasm. See, everything's kind of connected. And that amino acid bumps in here, tRNA bumps in here, we charge it, goes off and does its job. I think I'm going to stop with this one now because then we're going to focus on, let's see what's coming up. Should we do protein synthesis all in one shot? I think we can do protein synthesis all in one shot, and then this will be a little bit better. So we'll come back and do protein synthesis in the next